Good morning, children. Welcome back to Biology class, Chapter 6 Molecular Basis of Inheritance Human Genome Project. The sequence of bases in DNA that determines the genetic information of a given organism. In other words, genetic makeup of an organism or an individual lies in the DNA sequences. If two individuals differ, then their DNA sequences should also be different at least some places. These assumptions lead to the question of finding out the complete DNA sequence of human genome. The entire DNA in the haploid set of chromosomes of an organism is called a genome. In human genome, DNA is packed in 23 chromosomes. Human genome contains about 3 into 10 raised to 9 base pairs. With the establishment of genetic engineering techniques, where it was possible to isolate and clone any piece of DNA, and availability of simple and fast techniques for determining DNA sequences, a very ambitious project of sequencing human genome was launched in the year 1990. Human Genome Project was called a mega project. Why it is means human genome contains approximately 3 into 10 raised to 9 base pairs, and if the cost of sequencing required 9 billion US dollars. If the obtained sequences were to be stored in a type form of books, each page of the book contained 1,000 letters and each book contained 1,000 pages, then 3,300 such books would be required to store the information of DNA sequence from a single human cell. Okay. Next, we will discuss about the goals of Human Genome Project or HGP. First point, identify all the estimated genes in human DNA. Okay, identify all the approximately 20,000 to 25,000 genes in human DNA. Second point, sequencing of 3 billion chemical pairs of human DNA. Determine the sequences of the 3 billion chemical base pairs that make up human DNA. Third point, store this information in databases. Fourth point, improve tools for data analysis. Fifth point, transfer related technologies to other sectors such as industries. Last point, addresses the ethical, legal and social issues ELSI that may arise from the project. The Human Genome Project was a 13-year project coordinated by the US Department of Energy and the National Institute of Health. During the early years of the Human Genome Project, the Wellcome Trust UK became a major partner. Additional contributions came from Japan, France, Germany, China and others. The project was completed in 2003. Goals of HGP we are completed now. Now next it is what are the methodologies? The method involves two major approaches. First one it is the expressed sequence tag, focused on identifying all the genes that are expressed as RNA, ESTS. Second one it is the sequence annotation, sequencing whole set of genome containing all the coding and non-coding sequences and later assigning different regions in the sequence with the functions. So these are the two methodologies using in the Human Genome Project. Next, what are the procedure to using the methodology. First, isolate DNA from a cell. Isolate DNA from a cell. Second, it is convert into random fragments. They are the DNA selecting from the cell and it is convert into the fragments, dividing into the fragments, and then clone in a host or the vector. Using the vector is bacteria or the yeast. That vectors we can call bacteria artificial chromosomes or the yeast artificial chromosomes, and using for the amplification. Next step, sequencing of fragments using automated DNA sequence using the Frederick Sanger method. Next, arrange the sequences based on overlapping regions. Last point, elegant sequences using computer programs. See that one more time. Extract DNA, prepare hundreds of millions of genomic DNA fragments on a surface. Single genomic fragment, make thousands of capsules, genomic samples into sequencing machine, computational analysis of data. Okay, these are the process we are using for the procedure for the methodologies. Next, we'll go through what are the Frederick Sanger. What is that points? Sanger has also developed methods for sequencing of amino acids in proteins. DNA is converted to random fragments as there are technical limitations in sequencing very long pieces of DNA. Third point, Human Genome Project was closely associated with bioinformatics. Okay, what is that? Bioinformatics. It is the application of complete computer science and information technology. See, listen, application of computer science and information technology to the field of biology and medicine. That is called bioinformatics. Of the 24 chromosomes, 22 autosomes and X and Y chromosome, the last sequenced one is the chromosome 1 that was discovered on May 2006. Genetic and physical maps on the genome were generalized using information on polymorphism of restriction, endonuclease recognition site and some repetitive DNA sequences, that is called the microsatellites. DNA sequencing also have been done in bacterial, that is bacteria and yeast. Canoharbitis elegans, it is a free-living non-pathogenic nematode. Drosophila, the fruit fly, using in the plants, rice and Arabidopsis have also been sequenced. Now next we will discuss about the salient features of the Human Genome Project. Some of the salient features of Human Genome Project are the following. The human genome contains 3,164.7 million nucleotide bases. Human genome contains 3,164.7 million nucleotide bases. Second point, the average gene consists of 3,000 bases, but sizes vary greatly, with the largest non-human gene being dystrophin at 2.4 million bases. Okay, next point, the total number of genes estimated 30,000, much lower than the previous estimates 80,000 to 1,40,000 genes. Almost all 99.9% nucleotides are exactly the same in all people. Next point, the functions are unknown for over 50% of the discovered genes. Less than 2% of the genome codes for proteins. Next point, repeated sequences make a very large portion of the human genome. Repetitive sequences are stretches of DNA sequences that are repeated many times, sometimes 100 to 1000 times. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution. Chromosome 1 has most genes, 2968, and the Y gene has the lowest one, that is the 231. See that one, chromosome 1 has 2968 and the Y gene has that is the lowest one, 231 genes are present. The last point, scientists have identified about 1.4 million locations where single base DNA differences, that is called SNPs, single nucleotide polymorphism, SNPs, occur in humans. This information promises to revolutionize the process, finding chromosomal locations for disease associated sequences and tracing human history. Okay, what is that Human Genome Project? It is the mega project and it is identifying the human genomes. One cell contains the genome, that is the 3 into 10 raised to 9 base pairs are present, and it is connected with the what are the goals, what are the salient features, what are the methodologies are important. Thank you.